Good afternoon to all the participants. I'm glad to see such strong interest in the topic of nano-robotics and medicine. My name is Ilanit Mor. I'm head of the Industry Research Division in Infrastructure Division at the Israel Innovation Authority. As part of my role, I'm responsible for identifying emerging technologies such as nano-robotics in medicine, and promoting their development within academia and industry in Israel. You can see here the agenda of this webinar. We will begin with opening remarks from Dr. Aviv Ze'evi, VP of Infrastructure Division. Following that, Professor Gilad Yosifon will open the lecture series with an overview of the field. Then leading researchers and industry professionals in the field will present their work and discuss potential industrial applications. And Aviv, the floor is yours, please.
Thank you, Ilanit, and thank you for organizing this event. I just want to say a few words about the purpose of this specific event. First, I want to thank all the lecturers that came from the academia and the industry, but the main event actually is for the industry and for the academia. And we hope we will be able to create a meeting place for the academia and the industry on a specific technological challenge. And this technological challenge is nano-robotics. We already identified that there is a lot of knowledge that is done here in Israel, in the Israeli academia, but also outside of Israel, and we hope that the Israeli industry will be able to benefit it. So the main idea is that the academic researchers will present their abilities, and the Israeli industry will present their hopes or their demands. And eventually, post this specific event, they will meet and they will submit us a proposal. So I will not take more from your time. Thank you very much, and please continue.
Yeah, okay. So we'll start with Professor Gilad Yosifon from Tel Aviv University. Gilad, please.
Okay, thank you, Ilanit. Thank you, Aviv, for giving me the opportunity to give the first lecture. Let me share my screen. One moment. Does it look okay, or there are black boxes?
No, it looks fine. Thank you.
Okay, excellent. So thank you all. So this is a kind of an overview mixed with things that we did, research studies that we did in my group. So the topic is from micro nano swimmers to micro nano robots. We'll talk about fundamentals, the challenges, and some emerging opportunities. So I'm Gilad Yosifon in the School of Mechanical and Biomedical in Tel Aviv University. The video that you see is actually kind of a micromotor, a magnetic micromotor that we turned into a micro robot by adding real-time image processing. The yellow is actually the motion of the magnetic particle on the ceiling, and the cyan is actually the motion of the particle at the bottom. So it's kind of a 2.5 dimensional motion that we achieved. Overall, in the group, we are focused on micro nanofluidics, best lab on a chip. We are also focusing on micro nano robots, electrokinetics, and electronics as a recent topic in the last years. The motivation for untethered, and this is a crucial, important feature of tiny robots. So we start by untethered in contrast to tethered one. And actually that was kind of the vision put by Feynman on 1959 in his seminar lecture titled There's a Planning Room at the Bottom. And there was a follow-up movie, Fantastic Voyage, that actually talked about shrinking the surgeon in Feynman's visionary. Since then, there were many conceptual papers of how to actually introduce this kind of macro robots or tiny robots that can move in the body and can do different functions. We can also get inspiration from biological machines like the phagocytosis. And the benefits is that we are talking about remote controlled agents. These are able to undergo like an high precision controlled, minimally invasive, minimal side effects, and there is a kind of a large biomedical environmental potential to this. To start with the definition of how the macroscale robot, as we all know them, are compared to the microscale, then in the macroscale robot, we know that everything is integrated. We have all the components, what is called onboard components, controllers, sensor, CPUs, powering, everything. Now, in the micro robot, this is the micro robot. I hope that you can see the mouse. So the micro robot is too small, actually, to be able to have all the functionalities integrated on this micro robot. And then, actually, part of the components, of most of the components that are used for the intelligence and the control are actually off-board. And that's actually where comes also the definition between onboard robots versus off-board robots. And the macro robot, in this case, actually are treated more as an end effectors. So all the intelligence, all the control, all the vision is done off-board, while the macro robot has the minimal, most necessary functionalities. Here are different examples of such end effectors or micro robots that can be propelled in different mechanisms, chemical, magnetic, acoustic. Some of the potential application that can be done both in vivo and in vitro. Recently, since 2020, and I think that we have here actually both Itai Cohen and Professor Orlin Velev, people also started to integrate on the micro robot also some of the electronics. Okay, so it's not yet matured, but there are very nice efforts into making the micro robots also kind of more integrated. Okay, so this is a work from Itai. I will not take his time. He will explain. This is a diode that actually was studied by Orlin Velev, and the idea is actually to connect many diodes, many transistors, as much as needed, and then we have more autonomy. But again, it starts to appear, but it's not common, and not always it's beneficial. So we need both ways. I will take advantage. In the overview. I will use a recent ACS mega review. It's like more than 150 pages. Gladly, I'm part of the review, and also I can identify Professor Orlin Velev and many other review that was co-authored by Martin Pumera. And the review title is Technology Roadmap of Micro Nano Robots. And I will actually take you through this roadmap, and I will start by propulsion. So in terms of propulsion or microscale propulsion, we're talking about micromotors or active particles. There are many, many terms that people are using. And the idea is that at the low microscale size, actually the Reynolds number is very, very low. It means that the viscosity dominates. And we know that according to the scallop theorem, for a reciprocal motion like that of a scallop, there is no net motion. And biology knows it. And that's why different bacteria and biological agents actually use cilia, flagella, in order to result in a non-reciprocal motion and actually to have net motion. Now, when we as engineer want to mimic, of course, we will not, there's no way that we can really mimic the flagella or the complexity. So actually we get inspiration. And that's the way that actually we synthesize our micro motors or micro robots. We just break the symmetry and we use different energy or different propulsion mechanism, like the chemical propulsion, where we can actually generate a bubble due to catalysis only on one side, and then it will result in net motion. We can use magnetic, optical, ultrasound, electric field. We can also use biohybrid. The key is that the micromotor, or also the swimmers, or the self-propelling particles, all of them are the same terms. Actually, they are the building blocks for the micro-robots. Symmetry breaking is the key ingredient for the motion of such micromotors. Here you can see the Janus particle, the Janus, where you have only a catalysator like platinum, only on one side of it, or gold for electrical. So the idea is actually to turn the micromotor into a macro robot, and the way to do it is actually to integrate additional level of intelligence and control, like steering, interaction, smart interaction with the environment, closed loop feedback control, etc. There is also the possibility actually to use a hybrid powering, so we can actually take advantage of the hybrid powering to enhance the robustness of the operation. We can actually have a complementary actuation like propulsion and navigation, and there is also synergistic effect. I will show an example from my lab. So we did like electric and magnetic. You can see the motion of the Janus particle. It's about 27 micron in size. It can carry cargo on demand, but it was steered using magnetic field, but its motion is due to electric field. Okay, so this is one example. Another example is a micromotor that actually is rolling using a rotating magnetic field. So the rolling is magnetic, the navigation is magnetic, but the alignment is electric. Another example where we use electric and optical. Here we fabricated and engineered a double H-shaped micromotor, and we illuminated. Here you will see the illuminated using UV. And once we illuminate, the micromotor actually makes steered, it makes turn. So this is the way that actually we combine optical steering, electrical propulsion. We also did work with Joseph Wang, which unfortunately could not join us from UCSD, where we did even a triple hybrid, electric, chemical, and magnetic. And the previous work was together with Orlin Velev. In terms of collective behavior means that we have many of these particles that can interact with each other using different mechanisms. If we take the electrostatic force, for example, there is a very nice paper by Granick that actually shows that for many such particles, Janus particles, if you just change the frequency, you can really transition from different modes of a collective behavior. So the particle starts to feel their neighbors, and since each of these particles is autonomous, once they feel their neighbor, actually they form kind of a collective behavior, a very interesting and complicated one. So we also did such collective behavior. So we fabricated a semiconducting Janus particle, and we show that once we throw many of them, there's nothing. Now we turn on the electric field, and you can see that actually they self-assemble in a very nice way because there is a repulsive force. And once we turn on the UV, actually there is an attraction, and they change from something that is a gas-like, a repulsive, to a chaining-like behavior. The same can be done with magnetic field. This is from a work where we did with Tami Lotan, where we introduce magnetic particles that carry stinging capsule that can deliver some drug. And we carried all of them together to the cancer spheroid. And once they assemble there, actually we can see the injection of the drug into the organelle. Next, materials design. So this is taken from the ACS Nano recent review. So we can use different materials for chemical propulsion, for example, like zinc, like magnesium. For light propulsion, we can use different photocatalytic materials like titanium oxide, etc. For magnetic particles, we can again use nickel, ferrum, cobalt, etc., etc. Acoustic, we can actually break the symmetry so that we'll have like an impedance, acoustic impedance, and we can propel them. And of course, we can always use biohybrid. We can use from the nature different particles as templates to form our active or micromotor or micro robots. Again, it's too elaborative, no need to go into details. The only idea is to see that there are many ways to actually fabricate such micromotor. This is a bottom-up. There is also a top-down, and in the top-down, let me indicate the electrochemical deposition. Of course, the 2-photon, which is the nanoscribe, which has an amazing sub-micron resolution, and also, of course, the photolithography and the deposition technique. Again, some examples from my lab. So we use the photolithography. We are not limited anymore to the spherical Janus particle. We can actually fabricate any shape. We can also take sunflower pollens. We can take biological material like what we did here, and we can deposit gold on one side of it. And this way we actually broke the symmetry and we turned this pollen into a micromotor, into an active particle. The same we did with Joseph Wang. We could actually use the particle, the spherical particle, as a sacrificial template. And again, we could use atomic layer deposition in order to make like a multi-layer particles. Next, intelligence. So intelligence in the micro-robots, again, is inspired from what we know in nature. I mean, if we look at the human beings and we start from a single cell organism, we know that the complexity of the system is increasing, also the complexity of the nervous and the neural cells. The same goes with the robots. So if we look at the custom traditional robots, we know everything is integrated, but at the low scale robots, nano, micro, the level of sophistication, of course, is low. And this is why actually most of them are treated as off-board intelligence. So this is our micro robot. It repeats or emphasizes something that I said in the beginning, but all of this, acoustic, optical, thermal, all of these are external cues, external energy that can be used together with the control and intelligence in order to smartly control these end effectors. And of course, as I talked, we can bring more and more functionalities. We can actually introduce bioprobes and additional functionalities, but again, to a limited manner. And as a contrast, this is kind of an on-board intelligence. It's an illustration. It's not yet realistic, but it's also one of the trends that our people are trying to get into, is to bring some integrated circuit onto these microscale robots. And when I talk about microscale, in this case, it's like 100 on 100 microns as a typical size of such micro robot. Now, for our off-board micro robots, they are not stupid, because we can actually make them very sophisticated by harnessing the external off-board components and using a computer vision and AI and tracking algorithm. We already see many sophisticated ways to extract intelligence from these micro nano robots. For example, this robot that can actually solve a maze. We have like a supervised learning for autonomous navigation of a magnetic nano robot swarm. And we can also use a neural network actually to improve both the navigation and the tracking and identification, etc. In terms of off-board intelligence, we can also introduce and bring external hardware and software, as you can see here. So this is an example from my lab, where this is the micro robot, but we have the camera, we have the visualization, the optical path. We have, of course, all the processing, the algorithm, the tracking, and of course, we have also the magnebotics, magnetic field generator, together with electric field that can control our hybrid electrical magnetic robot. Again, we can perform this 2.5 manipulation, where we can actually shift the micro robot from bottom to ceiling. So this is the paper under review. It's already published on arXiv. We can also introduce a sophisticated optoelectronic trajectory, where we can illuminate our electrodes because the bottom substrate is a photoconductive. So we can actually program, we can do it in a very dynamic and reconfigurable manner. And we can actually form this kind of a sophisticated directed self-assembly of many micromotors into more complex micromotor, in this case, a microspinner. And again, this is a work with Orlin Velev, where we can actually use an optoelectronic closed loop steering actually to navigate our microrobot between these 4 waypoints. And the flashes that you see is actually a closed loop flashes that is done on the UV illumination in order to actually enable the precise tracking of our microrobot. Next, applications. So in terms of application, there are many applications. Most of them and the most mature are, of course, in vitro. And this is, for example, in vitro sensing, where the speed of the micromotor is in correlation to the concentration of the analyte. So this is one mode of sensing. Another sensing is actually to use our micromotor to do kind of a sandwich immunoassay with reporters. So we can actually do optical sensing once we actually bind to the target antigen. We can also do electrochemical sensing and actually take advantage of the fact that our micro robots actually can swim in the volume and can capture the target, whatever analyte we are searching for. Again, example from my lab. So this is one example where we can actually use the micro robot as a micro carrier. It can carry particles. We also did it for immunosensing. The particles are also functionalized against specific analyte. And the robots are such small that they have the precision of interaction with a single cell. So we also show that we can do a local electroporation, gene transfection, and electrodeformation of a single mammalian cell using our micro robot. Again, environmental remediation, I think that Orlin Velev will talk about some of his group, so we'll really not get into these details. But again, we can use this micromotor to target different pollutants like pathogenic microorganisms, oil spill. The advantage of using microrobots is the fact that they swim. They can actually do a very efficient way for searching. We can make an eye surface area. We can bind the enzyme for degradation, etc. In terms of the application, yes.
Yeah, we are short of time, so
3 minutes and
try
finish.
to conclude.
3 minutes. So the in vivo is two special challenges: imaging and remote powering. Let me skip all of this in vivo application and just show the challenges. One of the challenges, especially for magnetic field, is actually to increase the efficiency and the strength of the magnetic field. And this is why, in parallel to the micromotor, there is a huge effort in making this magnetic field generator better, penetrating better. Again, visualization in vivo is a huge challenge. Let me skip all of this. There is a trade-off between spatial resolution and penetration depth, and this is the summary. So I would like to summarize and say that, again, 1959, that was the vision of Feynman. Since then, the propelling was advanced. In 2015, also simple in vivo delivery was advanced. 2020, we can see initial signs of integration of electronics. 2024, we can see actually the effort in field generators. And this is kind of a visionary an illustration that was taken from an IM system of how futuristic surgery in the clinic will be. Together with magnetic fields that can actually operate and control the end effector. And thank you for listening.
Thank you very, very much, Gilad, for a fascinating overview on the field. I would like to invite Professor Itai Cohen.
Ilanit, there is just one specific question to Gilad, if you can answer really quickly. Regarding what is the energy sources for the sensors?
So many. I mean, as I showed, I mean, you could use optical, you could use magnetic, but it really depends on biocompatibility, depends on the depth of penetration. For in vitro assays, you can use all of them. For in vivo, it's mostly magnetic, but you could use also near IR. It really depends of where and how deep you want to affect.
Okay, thank you. I just want to remind that all the speakers can answer some Q&As that are on the chat, so you are welcome to do so. I would like to invite Professor Itai Cohen from Cornell University. Itai, please.
Can you see the screen?
Yes, we can see.
Okay, so the idea here, as was mentioned, was to try to think about how to get on board everything. And so the idea is that you would have something like an electronic brain and ways to harvest power from light with photovoltaics and actuators. And the vision is that actually the electronics problem is solved, that we can now generate line widths down to 3 nanometers, and at a 10 micron footprint, you can actually do a lot of computation. Our challenge in my lab that we decided to take on was actually the creation of the actuators, the end effectors that allow the robot to convert electronic information into physical action. And the idea that we had was that instead of trying to miniaturize machines the way that we've been doing for a long time, is that we really need to reinvent the process of how we're going to shrink machines and how we're going to create them at this scale. So our idea is to essentially steal all the fabrication tools from the electronics industry. We know how to make very, very small feature sizes in 2D. And so the idea will be to create the electronics and then build the robot around it in 2D using all the lithography tools we know and love, and then fold the robot into 3D. So if you're going to do origami at the micro scale, you have to have really good paper, and you have to be able to fold. And both of those are big challenges that we've been able to solve. And so here is an example of our microscopic paper. So this is this is a 10 nanometer thin film, and we've grabbed it with an electrode. And if you change the voltage on the electrode, you can bend and unbend it. Okay, so that's what's going on in this video. So how does this work? You take 7 nanometers of platinum, and you passivate it on one side with an inert material. You then fabricate the actuator that you're interested in using photolithography. And if you take that actuator and you place it into solution and you apply a voltage onto the platinum, then ions from the solution will absorb onto the active surface. And what you'll end up getting is a stress that bends the actuator. Okay, so you apply a voltage and you get bending. You apply the opposite voltage, and now the ions come off the actuator. It comes back to its original shape. You put on rigid elements. Now you have an origami. And with this technology, we've been able to make a microscopic hexagonal fold, a Mura-Ori, and probably the world's smallest folding duck. And the size scale that you're looking at here, when these things are folded up, is around 40 microns. Okay, so about the size of a hair diameter in footprint. Now, as amazing as these are, and they really are amazing microscopic origamis, you still need to actuate them using a wire from the outside. Okay, so all of the bright yellow elements here, they're all connected to this wire. You apply the voltage. If the platinum is on top, you get downward bending. If the platinum is on the bottom, you get upward bending. But I want to get rid of this wire. I want to do all of the actuation on its own. So how do you do that? You take photovoltaics and you connect them. So these are little solar panels that you connect to the actuators. And then you can shine light on your photovoltaic. And this is what you get. Boop, 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 boop. So you shine light, and every time you shine light, the actuator moves a little bit. And so now you go crazy. You put photovoltaics on the front legs, photovoltaics on the back legs, and you make bajillions of them on a single wafer. You put on the pads to restrict the bending. And then if you work hard, you can get this robot. This is a 40 micron by 70 micron by 2 micron robot. You shine light on its front photovoltaic. You actuate the front legs. Back photovoltaic, you actuate the back legs. And you march this thing along. And this thing is so small that you actually have Brownian particles here next to it. And this robot, for a while at least, held the Guinness World Record for the smallest walking robot. We have smaller ones now. As amazing as this robot is, it's still just a marionette. In a marionette, you have strings pulling the arms. Here we have lasers that are being directed at the photovoltaics. How do we cut the strings? So for that, we need an onboard brain. And so we designed a circuit that allows us to essentially drive voltage pulses to the legs of the robot at particular times in a cycle. And so if you put that circuit on, that's the central element here, then you can create AntBot. AntBot is a fully autonomous There's no strings attached. You just have a brain that's sending the signals to the legs. It folds itself up, and if you shine light on it, it just walks off the page. All right, so this is a integrated electronic robot where everything is on board except the power source, which is light. So we have been working on a number of different systems. So we have implantable electronics. We're making artificial cilia. Cilia are the little hairs that move mucus up from our lungs. I've told you a little bit about the microscopic robots. We're making metamaterial robots. But I want to tell you a little bit about how we're going into the realm of surgical tools and a little bit about actuators that work in air. So let me start with surgical tools, just a few minutes here. So what's the problem? The problem is if you want to make something that can actually cut or deform tissue, it needs to be a thicker film. 10 nanometers is not going to cut it. But if you make the film thicker, so in this case, the platinum is only being driven by the absorption of the ions on the surface, or maybe even oxidation, and that only affects the surface of the actuator. So as you make it thicker, the bending radius becomes much, much larger, and that makes it no longer a microscopic actuator. It means your robot is going to be much, much larger. So we had to come up with a different material, and one of the materials that we came up with is palladium. Palladium is a bulk absorber of hydrogen. So now the idea is that you put a voltage on, you absorb hydrogen into the palladium, and you stretch the palladium. If you take the hydrogen out, the palladium shrinks. And so that's how you can get actuation from a bent to a flat state. And now, because the palladium is a bulk absorber, you can maintain your radius of curvature even though your film is thicker. And with this, we've been able to create actuators that have anywhere from just 10 nanometers to 200 nanometers. And that allows us to operate in tissues like this. This is a tissue phantom. It's agarose gel that has a modulus that's equivalent to brain. All right, so now you can actuate inside of brain tissue. If you did this with a platinum actuator here, this thing wouldn't be able to move at all. So this is what it looks like. You have these rigid panels, you have these pads, these actuators that are driving the motion. And so this could be a little gripper that you can imagine doing a biopsy inside the brain. And in order to get it inside the brain, you want to link it up. So first, let me say that our actuator here is optimizing low operating voltage, high energy density, and a very small bending radius. It's this red over here. And these are the other sort of technologies that are available, and we're sort of sitting at the optimal corner of these technologies. The purple is also our technology. That's the platinum. The red is the palladium, so it allows you to operate in the body. So we're now thinking about putting these end effectors on the ends of fibers, trying to shrink our surgical instruments from something like this to maybe an optical fiber with an end robot. This is our first ever device, so we're still working on this. You have an optical fiber. It's about 200 microns in diameter. It has wires, and it connects to the robotic end. And if we apply a voltage, you can see that we can now actuate the ends of our robot element through the fiber. And so the idea here is to create a very, very small biopsy tool, about 10 times smaller than what is currently available, in order to miniaturize a surgical tool. We can also do things like make microscopic robot arms. And so this is an example of how we create multiple origami hinges to allow the robot to swing, lift, and twist, as well as grasp and manipulate the materials that it's grasping. All right, so all of this is done, fabricated in 2D, and then lifted into the third dimension for operation.
Okay.
So, yeah.
Yeah, please try to conclude.
Yeah, and just the very last thing that I want to mention is that we can now do this all in air and vacuum. So in this particular actuator, we have 2 platinum electrodes and an oxide in between. By applying a voltage across the electrodes, we can drive ions in the oxide to one side or the other, which allows you to bend one way or the other. And that's the what allows us to operate in solvent, in air, or under vacuum. And I'm going to skip this story here. this actuator is really fun. We're trying to make wings with it, but I'll sort of end by just showing the wing operating in air, operating at around 600 hertz, and say that these actuators can operate all day, no problem, when they're being driven in this way. So these robots, I think our idea is to essentially miniaturize and do everything that robots do at the macro scale, but now at the micro scale. And this idea of getting electronics on board is, I think, within reach, and it will allow for new functionalities. It'll allow for the robots, for example, to sense their environment and change their interactions with one another to create new emergent behaviors that you couldn't get when the functions were just static. And that's going to allow us to do all sorts of things like reorganize the spaces that they live in, shrink these surgical tools, get cargo transport, and really explore the microscale in ways that we couldn't do before. So I'll stop there, and I'd be happy to take any questions.
Thank you, Itai. Amazing. You can answer a Q&A in the chat, so please have a look. We'll go to Shai Poliker, managing partner at Edge Medical Ventures. Shai?
Yes, 1 second. Can you see my screen okay?
Yes,
Yes, it's okay.
Okay, great. So Edge Medical Ventures is it's a medical device venture capital fund and also a venture studio operating under the license of the Israel Innovation Authority. Basically, what we do is we start from an unmet need that we get from the industry, and we form around it engineering, clinical skills to be able to solve that. And I wanted here to give you an example of one problem, important problem that ended up being solved by nano-robotics. But as we always do, I will start from the unmet need. So this is a little bit of clinical. We heard a little bit of engineering here, so a bit of a clinical need. We are talking about implants in the body that sometimes get infected. It's a major problem. As you can imagine, it's very hard to treat with antibiotics. Implants do not get blood supply. It's very hard to reach. You cannot just open the body and clean them. And the main issue is a biofilm that is, encapsulating the bacteria and making this very hard to access. The initial problem that we try to address is infections that happen after total joint replacement. For example, after total knee replacement, where you can get the patient back after a few weeks with a severe infection. The rate of this happening is relatively small. We're talking about 1 or 2% of the procedures. But when it happens, it's a disaster for the patient. It's a very high cost and with horrible impact on the patient. And there's been a ton of attempts to resolve the problem. What they do today is open the knee and really try to scrub it and remove the biofilm. Extremely important. There's always a fight on how powerful your disinfectant can be without damaging other part of the body. So the way we tried to address it is by using metal nanoparticles that are biocompatible and basically act as sanding and removing of biofilm. So I think this was discussed before. This whole idea of swarm particles that are in nanoscale and are impacted or affected by different types of energy, pH, temperature, sometimes time-dependent change, and in our case, ultrasound. So what we combine basically is a solution with nanoparticles that is placed in the area of the implant, and then activated or stimulated by ultrasound. The big advantage here is the precision and the access. So one of the problems if you think about an artificial knee implant, is that you get all of these small cracks and areas that is very hard to reach. Sometimes you cannot even see them when the knee is open. And when you have a solution with nanoparticles and ultrasound, the traveling of the ultrasound is excellent through the fluid. This means that the nanoparticles will get everywhere where the solution is and can really produce powerful cleaning effect. This is how it works. You have basically two parts of the system. One part is the fluid, a simple cell line, which is filled with, as I mentioned, biocompatible iron oxide, simple nanoparticles with some certain shape, and an ultrasound transducer operating in levels that are known to be safe. And this create cavitation bubbles that in turn stimulate and trigger the nanoparticles and remove the biofilm. The device is already built, tested in animals, and towards the first clinical trial, you place this device just on top of the knee. You fill that with cell line and nanoparticles that are mixed in real time in the OR. So the activation of the transducer and the mixing and preparation of the solution all done in the same device in a very, very simple procedure. And then there is a session of a few minutes where the ultrasound is being applied and the nanoparticles are activated. And then you remove the solution with the nanoparticles and the kind of whatever debris that is out there coming out of the knee. What we were able to show while comparing this to the two most common disinfection solutions that have their own side effects and issues, and we show that we are significantly better than those, and also compare that to the ultrasound on its own, because the idea of using ultrasound for cleaning, this is it's a known concept. The combination, what we have found is that the combination of ultrasound and nanoparticles is really a powerful solution, and we're able to remove the biofilm without impacting the gentle coating that you find today on the implants of those of the joints. This is, I will try to play the YouTube video. I don't know if you will be able to see it. Can you see it?
Yes.
You fill the device with saline. Particles, there is a mixing of 2 minutes. You take out the mixed solution. And few minutes of activation is already quite effective. This is less than 10 minutes. So,
Okay, Shai, please please try to conclude.
Yes, no, this is so I think that the main thing here, I think in terms of the technology around this is much simpler than I'm sure most of what we heard so far and we're going to hear, but it's important to see that these things have very practical and real clinical applications, and we are looking forward to use more of those in the future. Thank you very much.
Yeah, thank you, Shai. Yeah, this is the idea. I hope that some collaborations will come out of this webinar, bringing more of academic research to the industry like this. And with that, we'll move to Professor Izhar Or from the Technion. Izhar, please.
Hello, can you hear me? Can you see the screen?
Yes, just move to presentation mode.
Okay, now you can see?
Yes.
Perfect. So thank you for the organizers for inviting me and for the speakers' wonderful talks so far. So I'm working on theoretical analysis of nonlinear dynamics and mechanics of under-actuated robot motion. And in particular, here I will present works relevant to robotic micro swimmers. So Gilad mentioned the wonderful introduction, the speech by Feynman, and also the scallop theorem. The scallop theorem was mentioned, coined by Purcell. There is a wonderful speech and paper, Life at Low Reynolds Number, 1977. It's almost 50 years ago. Very classic, recommended reading. Inspired by motion of swimming microorganisms with waving a flagellated tail. And Purcell considered this simplified model, if you can see my mouse, which is trying to mimic the undulation of a tail, but with a discrete minimal model of 3 links and 2 joints. Of course, because of scallop theorem, we need more than 1 degree of freedom actuated. So in this model, the relative angles phi1 and phi2 are prescribed kinematically by the robot, by motors, by servo motors, or any other actuation way. And doing something periodic in the plane of these angles generates some loop. It can be square-shaped, it can be circular, it can be ellipse. And the nonlinear dynamic equations for this swimmer were formulated only 25 years after Purcell in 2002. Wait, I'm hiding the bar. Okay. Most importantly, like Gilad mentioned, is that this is a low Reynolds number swimming, which means that we neglect inertia. So we assume quasi-static motion, 0 total net force and torque. And from this one can obtain equations of motion, a kinematic system, first order ordinary differential equation system. I will not go too much into the math here. And in our work, we did some analysis of this model and also built robot, but not micro robot. For the first thing, we built prototype, which is macro scale. And in order to have dynamic similitude, so we work in low Reynolds number, instead of using water, we used the highly viscous silicon fluid. It is 60,000 centistokes in viscosity, while water is 1 centistokes. So it's very viscous. We were able to show, for example, that if you make this input, which is sine and cosine, which is drawing a circle in the plane of the 2 angles, and changing the amplitude, the stroke amplitude, there is an optimal amplitude that maximizes the displacement that the robot is moving per cycle. And the discrepancy here is between the model, theoretical model, which assumes very thin rods, slender links, and the robot, which is actually not really slender links, and it has another flotation foam in the middle, and then we used a simplified model for this. And later we were also, this is another untethered prototype, and with a different input, we were able also to show a reversal in the direction of motion when we change the amplitude of the stroke. for about 90 degrees. But all this is a robot. We also did some works on optimal control for this. All this is just a model robot in macro scale and with actuation of motors. When you go to micro scale, you cannot do this, obviously. And I'm not an expert in experimental work of micro swimmers. In that case, I cannot show the wonderful works shown previously by Gilad and by Itai. So I collaborate with people who do this. So this was the first micro swimmer introduced 20 years ago. Very famous. It's a work of Dreyfus, Howard Stone. There is beads, superparamagnetic beads, and there is an oscillating magnetic field which is causing this to move. And one interesting thing that they found is when they increase the oscillation amplitude, suddenly this swimmer starts to move in perpendicular direction. It moves in a direction which is perpendicular to the mean magnetic field. The magnetic field is constant in X direction and oscillating in Y direction, and they have this change in the direction, which was kind of unexplored further. I was working later with groups like Brad Nelson from ETH, who was fabricating much simpler structure of a swimmer, which is just, so to speak, a two-link swimmer, a nickel ferromagnetic head with a flexible hinge, semi-flexible tail, and our theoretical model is just two links with a torsion spring. We are able to formulate the dynamics of this swimmer. Similar equation, that's the torque generated by the magnetic field, trying to align the links with the magnetic field. It's proportional to minus sine gamma. That's the angle between the direction of the magnetic field and the magnetized link. We were able to formulate the dynamics, apply some analysis to find optimal actuation frequency, optimal stiffness. Here, the motion is not time invariant, so we have optimal frequency, which maximizes the speed or the displacement per cycle. Then we went to a small modification of this to try to explain the jump in 90 degrees, the motion change in the direction from motion in X direction to Y direction. We considered this simplified two-link model, but now the head link is super paramagnetic, which changes the expression for the magnetic torque, now proportional to sine 2 gamma, which means we have equilibrium also in perpendicular orientations. Then we did some mathematical analysis. I will not go into the math. If this is interesting, I can explain later privately. And we were able to show why this swimmer is actually flipping from motion in X direction to motion in Y direction. We collaborated with a group from Chinese University of Hong Kong who fabricated this swimmer. They constructed also the array of the magnetic coils, and under the microscope, they can see this. So for small amplitude, the swimmer is moving in the X direction with optimal frequency. When we increase the frequency, we can get this transition not just by changing amplitude like predicted previously, but also by changing frequency. You can see when the frequency is increased, this micro swimmer is moving and transitioning to Y direction. And we were able to do the analysis and to match between the theory and the experiment. So this was a wonderful collaboration, not with industry, with academia. I didn't have time to explain what is the motivation for doing this for biomedicine, but obviously it was explained previously, targeted drug delivery, cargo moving, biopsies, taking a small camera for inspection, and many other motivations. One more thing is for helical swimming. So this was fabrication of nanohelix with a nickel head. And later we discovered that it is much simpler to use this fabrication of the two-link swimmer and putting it in a rotating magnetic field, which is rotating in the plane. And you have very interesting dynamics, which depends on the frequency. If the frequency is too low, the swimmer is just tumbling in the XY plane, the plane of rotation of the magnetic field. If we increase beyond some critical frequency, then it starts moving in a helical path out of plane in the Z direction. Then you have a maximum, and at some point there is a phenomenon called step out, in which the swimmer loses synchronization with the magnetic field. And all this we were able to predict with a very simple theoretical model of the nonlinear dynamics. I think I will have to conclude. Yes, Ilanit. Okay. Yeah. So just last slide, I mentioned some collaborators, and I hope that this wonderful workshop webinar will stimulate more collaborations. Right now, I'm from Technion, and I'm also representing Professor Alex Leshansky. He's now in the audience. We are both doing theoretical and numerical analysis of microswimmers. My collaborators, I mentioned Professor Brad Nelson from ETH Zurich, Li Zhang from Chinese University of Hong Kong, Min Jun Kim from Southern Methodist University in Texas. He is also a collaborator with Leshansky. They received a nice grant, NSF-BSF. Now I applied for a joint NSF-BSF with Min Jun Kim. Leshansky has also collaborators, Pierre Fischer from Germany, Ghosh from India. He has a startup, but we are seeking also for interesting collaborations from the industry, also in Israel, and also academic collaboration with the wonderful speakers that showed wonderful presentations before, and probably also after. We see that Professor Velev and many more will also speak today. We co-organized a nice workshop on microswimmers and soft robotics at the Technion in 2020. That was right before the COVID. So I hope we will be able to collaborate and organize more conferences and more collaborations between academia, experimentalists and theoretical, and also between academia and industry. Thank you very much.
Thank you, Izhar. If any help is needed further to organize more sessions, we will be happy to help. And with that,
thank you.
Thank you. And with that, we'll move to Professor Orlin Velev from North Carolina State University.
Okay, you can ask questions in the chat. I'm muting now.
Thank you.
Okay, I hope you can see my screen and my presentation.
Yes, we can. Thank you.
Okay, so it's really a pleasure to participate. For me, this is early morning, but the topics are so exciting that it's really very nice to be connected here. What I'm going to do is I'm going to focus on one of the specific aspects that relates to microrobotics, and this topic is self-propulsion. So when it comes to the background, Diewald already showed one of those beautiful images. But this topic has been a big interest and inspiration to society in general, just because of the mass media movies such as the Fantastic Voyage, which was mentioned. There is another movie that I would recommend to movie fans, which is called Inner Space, which is actually guided by Steven Spielberg at the time. So these are really inspiring type of course presentations, but the question is, how do you make a device like that? And even harder is, how do you make them move? And there are two big problems. So even if one achieves exactly this problem, there are two big challenges in front. One is Brownian motion. We kind of won't be discussing this too much here, but the other one is, how do you make them move on their own? And in order to delineate what I'm going to be presenting here, I'm really going to kind of separate the types of motion that one can see on the micro scale in present day micro-robotics and nano-robotics in two categories. One is the so-called theoretically driven particles. This is the case where we apply external field. The particles are driven by the field and can make the directed motion. This is a technology that by now has been maturing quite a bit and has found applications in a number of biomedical devices, which I believe are more or less at development and testing stage, as kind of like the devices that use multiple magnets and biomedical imaging in order to move micro robots inside the body, for example. I'm not going to discuss this technology too much. I'm going to discuss even though this, of course, technology right now, the kind of the theoretic driven motion is kind of like getting commercialized. I'm going to discuss the challenges in front of self-propulsion. That is, how would the particle move on its own? The motion in this case has to be induced at the particle level. So the gradient that moves the particle needs to be a gradient that is being generated and focused along the particle. So how can I achieve this? I'm going to illustrate a number of approaches. Just before I do that, I want to kind of give a little bit of historical kind of flashback to the field. I'm still happy to be at a conference in 2000, around 2000, when some colleagues from Penn State University were reporting a kind of strange phenomena, which was that bimetallic particles move in hydrogen peroxide. And hydrogen peroxide since then has been one of the favorite media for moving such particles. Since then, the field has grown in two directions. And probably this makes this webinar specifically important at this stage, because the field has grown in terms of making self-propelling particles which are more complex and looking for applications which are more focused. At present day, we saw some beautiful examples of really complex systems that can be made in the presentations, and also obviously biomedical and similar type of applications, including environmental applications, are kind of emerging as one of the coming topics. So let's kind of go and take a look at a few examples of how one can achieve the propulsion on the micro scale. So the scallop theorem was mentioned. You may try to do mechanical propulsion, but that won't work on that scale because of the laminar flow. Basically, kind of like the general explanation about this is that this is really propulsion if you scale up everything to our scale. This is propulsion in something which will be the density of honey, or kind of like in the area where I live, kind of like, this will be kind of like, basically hot syrup. But either way, it is very hard to make mechanical propulsion. It is possible, but one of the simplest ways of doing this, which we have been investigating some time ago, also in collaboration with our Israeli co-workers, is electrohydrodynamic propulsion. This is an example of our work years ago, where one can apply external electrical field, which is AC field, and in this way it can penetrate liquids and induce electrical charge. So this is a microscopic diode. In this case, the scale is about a millimeter size, which is propelling by an external AC field. The power of this method is that one can scale down the propulsion to the micro scale and even nanoscale level. One can make electrically functional particles, such as the microdiodes shown here. In thinking about the scale of electrical engineering technology, one can really make all kinds of smart circuits on that scale. And here is an example why we call this self-propulsion. These are two microdiodes which are actuated by exactly the same field, yet they move in different directions just because they are facing different directions. So each of those microdiodes converts the external electrical energy into directed motion and moves along its own trajectory. One can combine such type of motion with other type of field-induced interactions. So here are two microparticles that can either be assembled at a certain frequencies, that is being attracted to each other, then disassembled and then can move, and then assembled again. And here is a beautiful example of what we would call synchronized swimming of particles, which are both driven by this type of field, and each of those creates its own driving force. So this is a very interesting topic. Now, the future vision was already pointed out in the talks before me, so I'm not going to go into too many details, because again, how one achieves this type of integration and what it is used for is like still an open challenge. But there is a clear path to devices that can be controlled and powered by external fields, that can move on their own, that can contain a smart circuit, can contain their own sensing, and typically, given the potential cost and complexity of such devices, would have functionality that relates to biological applications. So this is when it comes to electrical fields. Now I'm going to go actually to simpler and simpler type of systems, just illustrating something that is kind of possibly closer and closer to applications. Magnetic fields were mentioned, and the previous talk actually had this excellent example of the so-called helical propulsion in magnetic fields. Again, we're not speaking of particles that are just going to be driven by the magnetic field. We're speaking by particles that are going to use the magnetic field in order to push themselves around the surrounding fluid. Here is an example of a magnetic microbot, which is assembled from 4 microcubes, which have a magnetically actuated side here. It's not seen on that view, but basically you can use external field, move that small microbot, capture live cells, and then move it further. If you notice how this works, this is actually theoretically driven microbot as we pull it along. But the point is that if we actuate microparticles on the micro scale, we can also use the propulsion against the fluid around them. Now, it was mentioned that one cannot make a scallop that is going to be propelling itself in normal fluid on that scale, but something that has been shown earlier, and we have demonstrated also in our work, following the work of researchers such as Professor P.A. Fischer in Germany, is that one cannot make a scallop or artificial fish that is going to be moving by opening and closing regardless of the rate in normal fluid because of the viscosity, but one can move self-propelling particles in non-Newtonian fluids. And non-Newtonian fluids, by the way, happen to be also most of the biological fluids. So happily or not, such type of magnetically actuated self-propelling devices can really operate in biological fluids. This is an example of a microbot that moves in shear thinning fluid that we have reported some time ago. This is an example of some research which we hope to publish very soon of how the use of non-Newtonian fluid can lead to total reversal of the mechanical actuation on the scale, or can lead to the self-assembly of particle complexes that then can move on their own, such as this car-tool type of self-assembly. So this is the magnetic actuation. And I want to give a few examples of the simplest way of actuating particles, which is using, in this case, chemical gradients. I mentioned the hydrogen peroxide. A much kind of like faster way of moving particles would be to create gradients on surfaces of the interfacial tension, which is called Marangoni effect. And then the interesting type of collective phenomenon we mentioned. We can use the Marangoni propulsion.
Orlin, please try to conclude because we're out of time.
Yeah. Okay. So we can use Marangoni propulsion in order to move particles on top of fluid. We have recently demonstrated how this whole cycle can be applied to microcleaners that would self-disperse, sink down, capture microplastics, and float up. We have demonstrated how one can use particles that just dissolve directionally in order to potentially apply similar type of processes to disinfection and bacterial field disruption, another interesting problem that has been mentioned before. So I would like to conclude here, actually as a final slide, which is not directly relevant. I would like to acknowledge the long-time collaboration with our Israeli colleagues, Milat and Tuvia Milo, on fundamental topics, which I won't be discussing here, but we recently celebrated Tuvia's 80th birthday that I was really very glad to be able to visit Israel last week. So thank you very much, and I'll be glad to answer questions in the chat.
Thank you very much, Orlin. We'll move to Dr. Ronen Ivry, CEO and co-founder of Barcode Nanotech.
Thank you very much. So Barcode is a company in Yokneam in Israel. We were established in 2017, and we are actually developing drug delivery vehicles. But if you think about it, we're developing nanoparticles that can take different nucleic acid-based therapeutics these nucleic acid sequences could also be the basis for development of new types of self-assembly robots, if you want, or sequence different proteins that can then be in the target cells, decoded to become, if you want to call them, biological robots. So the challenge behind delivery of RNA or DNA or any nucleic acid sequence is how to get them into the right cells. And this is a fundamental question that Barcode is trying to deal with. So in order to solve the challenge of delivery, we've generated a platform that can allow us to identify nanoparticles that we build on our own that can lead the RNA or DNA sequences to the right location, the right target, once they are injected systematically. We've successfully demonstrated nanoparticles that can target areas outside of the liver, which is the largest challenge for nanoparticle delivery, how to get away from liver delivery. And we have generated thousands of proprietary new lipids. So we are not only generating the nanoparticles, but we are generating new types of lipids, the chemistry behind new molecules that can help us guide these nanoparticles better. And I'll tell you a bit about that. We already have strategic collaborations with biopharma companies who are looking for the optimal delivery vehicles to each target cell. And the IP and the company is actually a spin-off of the Technion. So the challenge that we're trying to solve is how can you deliver these nucleic acids to different types of cells. And the nanoparticles that we design are actually lipid-based nanoparticles. They are composed of different types of lipids, meaning that they can interact with the biological membranes, and with the idea that if you build the right component, if you build the right nanoparticles out of the combination of the right components, you can actually generate a nanoparticle that is target specific. And this is what we do. So the challenge is actually screening through huge number of nanoparticles compositions, because if you do the math here and you have all these different components built in together into a nanoparticle formulation, the number of possible formulations is a very large number, and that is the challenge we're trying to solve. So how do we do this, and how does our platform work? We start with the lipid chemistry. We design new types of lipids that go into the formulation using AI-guided generation. So we're taking databases that are from either published work or from databases that we generate on our own using the screening method. We then generate these lipids. We place them into an LNP formulation. And here we use another AI-based selection tool that we've built in-house that can help us build the different nanoparticle formulations. So each experiment that we run with a number of at least 1 million different nanoparticle formulations. Then using design of experiment tools, we move into the third phase, and we don't actually go into the lab and generate 1 million different formulations, but in a given experiment, we can generate hundreds of different formulations. And what's unique about our system is that we can place a molecular barcode to tag each type of formulation. So we can inject simultaneously hundreds of different formulations into the same animal as a screening model to understand the biodistribution profile of each formulation. So that is how we generate huge amounts of data with a limited number of animals using a huge number of nanoparticle formulations. Now, using this platform, we can identify the optimal nanoparticle delivery vehicle for each target, and we can generate feedback optimization using the data that we've generated to optimize the lipid chemistry of these components and to optimize the nanoparticle formulation. And when we inject the nanoparticles, in this case into the bloodstream of the animal, we can identify the delivery in each given target. So in this case, for lung tissue, we can take out, after injecting a mixture of 100 different nanoparticles, each one with a different barcode, we can quantify how many of that specific nanoparticle formulation, how many nanoparticles have reached the lungs. And then we can plot this and compare the delivery of the different types of formulations. So here, each bar in this graph represents a single LNP formulation, and the y-axis is actually the number of barcodes, the number of which represents the number of nanoparticle formulations that has reached the liver, in this case, the lungs. And therefore, the top formulations on the right-hand side are the formulations that better target the lungs. And because we're screening through all of the different organs and tissues in that same animal, then we can look at all of the different organs together and identify the optimal formulations that reach each organ or each tissue in this case. And this also enables us to perform another analysis. And this analysis is actually for each formulation. So if you look at a formulation and you're trying to take it away from the liver and into other types of targeted tissues or organs, one of these very important organ is for therapeutics in this case is cardiac delivery. So we can, using an optimization process, drive our formulations by understanding the different components and the different parameters that affect the delivery into the heart in this case, to optimize delivery into the specific tissue. And this is how we do the optimization process. So if you look at this from the nanoparticle perspective, we can start with, on the left-hand side of the slide, we can start with one formulation that once injected into the bloodstream can lead the nanoparticle into the spleen and liver. So if you see on the left-hand side, this nanoparticle is mostly going to the spleen, but it also goes into the lungs. And in a first iteration between these formulation changes, we change different parameters in this nanoparticle formulation to drive this formulation through this design to go more into the lungs than into the spleen. And then in a second iteration, we are able to increase delivery into the lungs by understanding what are the components that make this happen better. So through these iterations, we can make better and better nanoparticles to reach these tissues. I will not go into the numbers, but I can say that in terms of our ability to screen, we are doing this much faster and much more efficiently than any given company in the world right now that actually generates these types of nanoparticle formulations. And we already have some nanoparticle formulations that can target, once injected systematically, can target tissues like the lung, the spleen, of course, the liver. And we've been able to improve through our platform delivery of systematically injected nanoparticle formulations to the brain by more than 1,000-fold, to the heart more than that, and to the kidney, which is also a very challenging organ to reach with these nanoparticles. And what's important to this specific discussion here is that we can encapsulate growing numbers of nucleic acid sequences up to base editors. So meaning anywhere around 10 kilobase of RNA or DNA sequences, which can then be generated, as I said before, into biological so-called robots.
Ronen?
Yeah?
If you can, yeah, okay,
excellent.
Thank you.
Very strong IP held by the company for nanoparticle barcoding, so enabling the efficient delivery screening. And of course, this platform IP allows us to generate patents for new nanoparticles and new components. And I want to say that this kind of work is done by actually integrating, as people demonstrate here before, different types of disciplines, scientific disciplines. So here each team member brings her or his different expertise. So molecular biology combined with chemistry and analytics, with bioinformatics and AI, and of course with nanotechnology and formulation. And this is a team that makes this happen. So I want to thank you very much. And of course, happy to talk about collaborations and discuss them both with academia and with industry.
Great, thank you very much, Ronen. Yeah, we're a bit late, so I hope the audience will stay with us for 2 more talks. We'll move to Professor Slava Krylov from Tel Aviv University.
Okay. Okay, thank you, Leonid. So I will speak about micro-nano system technology, MEMS NEMS, as enabling technology for nano-micro robotics. I'm not a robotics person, but I think that this topics could be important. And what is good to be one of the last speakers, that a lot of things were already covered by previous speakers, and I will only touch few points that could be important in the development of future robots. So if you look at the robotic system, it has a lot of sub-functions, subsystems, and some of these functions are part of the platform. So robot has to have more locomotion to navigate, to keep a balance. On the other hand, some of the functions are related to functionality. For example, it could be drug delivery or surgical tool and so on. And the point is the MEM technology is enabling technology for many, if not all, of these functions. Of course, some of these functions could be onboard or off-board, but because the vision, as was already mentioned, that to reach the point where we have autonomous system, some of these functionality should be on board. And for this, we need actuation and sensing and mechanisms. These are things that I'm going to cover partially. So if you look at every basic electromechanical system and micro-nano-electromechanical system, the flow here is they have electrical signal, which is converted into mechanical system, mechanical signal, it could be motion or vibration and so on. And after that, the displacement is detected by another transducer, and there's a control. So these transducers, there are a very large variety of transducers. The most common in micro-nano world electrostatic, and all these things could be subdivided in 3 groups. It could be actuators which develop high forces but relatively small displacement. It's, again, the most popular is electrostatic in most of the MEMS devices. Magnetic is widely used for external actuation off-board, which was already mentioned during previous talks. And another group which is very relevant to the robotics, the elastomers, so electroactive ionic devices, which usually develop large displacement, relatively small force, and they're relatively small. Concerning the electrostatic actuation, is again the most common. It develops large force at small displacement. It also suffers from instability, so the design of these devices requires certain consideration to prevent these instabilities or analyze them. But this is basically behavior. The distance here just a few micrometers. Another approach is so-called area-changing electrodes, where the motion could be extended to a certain extent, but still they can suffer from instabilities. Can we see in here? So what I'm trying to say is that these things are apparently very basic, but every time we design the device, we encounter these issues which should be resolved and avoided. It represents significant challenge, and definitely in the area of micro-nano robots, if you want to design it. One of the approaches to partially avoid these issues is to convert high forces, small displacement to large displacement. And for this, motion amplifiers could be used. In the beginning of the MEMS era, MEMS literature, the literature was full of these images of gears and transmission lines, but very quickly it turned out they suffer from a reliability problem and stiction, because the surface dominating force prevents really free motion and increased friction. This is why today the trend that everything should be so-called compliant. So the area of compliant mechanism of motion and force transformers are very important for every mechanism, starting from biological system, insects, and also MEMS devices. Just a few examples of this compliant mechanism. So in our group, we developed with years several approaches to this motion transformation, motion amplifiers. This example of so-called negative Poisson ratio device, that's another amplifier. That what these things are allowed to do is to convert very small displacement of, for example, electrostatic actuator to large deflection or large displacement, which could be used for locomotion in micro robots as an enabling technology. Another group of actuators are polymeric actuators, which are very convenient for the use in robotic system that we already saw again in previous presentations. And one of the issues with these devices, first ionic devices are usually relatively slow, and this mismatch between the stiffness of the electrodes and polymeric material sometimes an issue. So again, we developed several approaches how to overcome this by using polymeric electrodes with collaboration with Yossi Shaham, or another kind of architectures which allows to extend the actuation range in this kind of devices, which could be again used in robotic application. Now, I will not extend about fabrication. What I will say is that in MEMS at least, the fabrication method is very, very established, and it could be manufacturable in industrial high volume fabrication, because not all the, research works and fabrication approaches could be implemented in the industry. And at least in MEMS area, there are a lot of approaches that could be manufacturable. A few examples of devices that we fabricated in our nano center here in Tel Aviv. Now, one point that I wanted really to address is the following. Robots, in addition to locomotion, they also need sensing. And if you speak, for example, about inertial sensors for guidance on control or stabilization or intra-body navigation micro robots, if you speak just as an example about accelerometers, so most accelerometers, they use static operational principle, just mass deflecting under inertial force, and this deflection is measurement. Another approach, which is very intensively researched now, are so-called resonant inertial sensors, where the principle is that changes the frequency of vibrating element under force. It's more or less similar when we tune the violin string, by pulling the string, we decrease or increase the frequency. And there's a lot of architectures of these devices which are used and also commercialized. The important point here, if you look at the trend of these devices, is now resolution up to nano G, with G's acceleration of gravity could be reached, but this is the only formula in my presentation. This is a formula also called noise equivalent acceleration density, which is basically the smallest acceleration which could be measured. And what happens here, if you look at this formula, we have this ampere temperature, but we have the mass as a denominator, which means that when we downscale the devices, the noise influence increases. And this is very key challenge in the development and further miniaturization of this kind of sensor, basically any sensor, that when we have smaller device, noise is more pronounced, and this challenge could be addressed. So it's a very interesting and important challenge if we can design really small sensors, for example, inertial sensors, to fit the nanoscale in such a way that it could be implemented into nanoscale robots. So it's just example of resonant accelerometers that we developed here, but it's not nanoscale, it's regular MEMS device based on this frequency monitoring principle, and it includes also some kind of motion transformation. It's a relatively complicated machine. And again, the vision that one day we will reach the point where we can be able to develop robots implementing this kind of complemented machines.
Slava?
Yes, that's one last slide before the end. So the last point, look at this, the size of these devices decreases steadily with time. And this is the last example of accelerometer from Bosch, which is 1 millimeter by 1 millimeter. MEMS-NEMS integration is very important. So what I'm trying to say is that right now, just to try to push down the scale of these devices, to make them less and less, smaller and smaller, technology for fabrication exists. This is 50 nanometers wide beam. We also use carbon nanotubes as a sensing element instead of silicon devices. So again, the future that we want to go is to continue this trend to miniaturize these devices towards the implementation in nanorobots. Thank you very much.
Thank you very much, Slava. We'll move to the last presentation by Professor Orit Shefi from Bar-Ilan University. Orit, please.
Yeah.
Orit, tell mute. אורית?
Okay, now I'll try to share. Sorry.
The share is working. We can see, but you're not
You can
in
see it
presentation
already?
mode.
Yeah, but it's
okay.
not, just not in the presentation mode.
Okay. I can see it. It worked before. I can see it.
If you have two screens, so probably you share the wrong screen.
No, I don't. Let's try it. Okay. Now it is okay. So thank you. I'll go through it fast. I'm not a nano-robotics or micro-robotics person, but we are in the lab working on nanoparticles, microparticles, and interfaces with biology that I thought it might be interesting and can be combined into micro-robots. I'll talk, the focus of the lab is tissue engineering and repair, mainly nervous system repair. I'll show you some examples of our research, and I hope you will find it interesting, and maybe we can find some collaboration. So there are three elements that I'm going to show you for remote stimulation or control or drug delivery via magnetic forces, ultrasound waves, and light. And these can be also combined together. So for the magnetic manipulation, we take two approaches. One, by developing nanoparticles for uptake. So the cells uptake magnetic nanoparticles and become magnetized. Also, we conjugate drugs to the magnetic nanoparticles and can remotely deliver them or target them into tissues. And the other approach is by coating from the outside biological elements. Here you can see some cells that uptake red labeling magnetic nanoparticles, and they are attracted to magnetic, according to magnetic flux, magnetic forces. And we can, with those, also we can interact from the outside elements, nano or micro scale magnetic particles. This is through biotin streptavidin, for example, that can be targeted. And we can also take particles and coat them with factors like growth factors or other drugs. I'll show you another example. And also can control their movement towards some biological targets outside and inside the body. Here you can see neural networks that we organized magnetically. You can see magnetic lines that we fabricated. These are devices that we are doing in-house and with some magnetic layers. This is in collaboration with Amos Sharoni from physics in Bar Ilan. And the magnetic forces are along the entire pads that you see, not just at the edges. So the magnetic forces are perpendicular to the substrate, and the particles that uptake, the green ones that uptake magnetic particles like here, aligned nicely with the magnetic elements. Here, they are not magnetized and growing everywhere. So this is one example. I'm moving to another, and also with the magnetic particles, we also organize organoids in 3D, in collagen. We can also control the environment of the cells and control, I haven't showed it here, but control the extracellular of cells and also collagen fibers orientation. So it's also a way to manipulate or interact with cells. This is another project. Here we made PDMS-based chips loaded with drug, and we showed that when we, with 20,000 kilohertz, we could make cavitation and a release of the drug. So the blue line shows a continuous release of drug, and in the green line, we actually activated the ultrasound only for 4 minutes. And what we've shown that we can do it in a repeatable way, and turn on and off the ultrasound, and after that, the release. So this is another project that we can activate or stimulate drug release. Here I want to show you another approach. We did it with gold nanoparticles that we coated with anti-cancer drug, the MTHPC, and we made a dual effect by light. We use 2 different light sources to go to photodynamic therapy and photothermal therapy, and we showed a synergistic effect. So, and also we could apply the photodynamic therapy via magnetic nanoparticles that we attracted to the tumor. Here, I'm not showing the result, but we injected systemically the magnetic nanoparticles, attract them with magnet, and then applied the photodynamic therapy from the outside. So this is another direction in the lab. Here you can see this mice that we targeted with the particles and activated the drug. And here is what I said about the magnetic nanoparticles. So you can see that here we have a tumor. We injected it systemically, and we could pull the drug carriers to the tumor and actually showed a very nice activation of the drug in a better way than without the magnetic nanoparticle, the magnet, sorry. These are the 3 directions that I wanted to show you: the light, the ultrasound, and the magnets. We have more projects in the lab, and I can answer and hopefully interact with some of you, which showed beautiful presentations.
Okay, thank you so much, Professor Orit Sheffi. We will now conclude. There are still some questions in the Q&A, so if the speakers can relate to them. I just want to thank you all for joining us today. We have had the privilege of hearing from leading researchers and industry professionals about very exciting developments in micro and nanorobotics in medicine. The insight shared today highlights the immense potential of this field and the importance of collaboration and also opportunities of collaborations between academy and industry. We hope this session has sparked some new ideas and will end in connections and opportunities for future partnerships. As mentioned, you are welcome to explore funding opportunities through our various programs, and we encourage you to reach out with projects, proposals, or collaboration ideas, and we will be happy to help. On behalf of the Israel Innovation Authority, I'd like to thank our speakers. We look forward to continue to supporting innovation and technological advancements in Israel. Wishing you all continued success, and we hope to see you at future events. Thank you very much.